Welcome back. What we're going to do in this video is we're going to talk about a few short mechanisms and get a little more clarity in some of the carbon dioxide transporters in the blood. Now, in one of the previous videos, we looked at and we said that in the hemoglobin molecule we have lysine residues, right? So 1, 2, 3, 4. Right? And we said that assuming lysine was deprotonated, right? Assuming it was deprotonated and we have a CO2, what happens? Well, if I load the blood up with CO2 by Le Chatelier's principle, you're going to consume the CO2, right? So nucleophilic attack, pi bond kicks up. What do we generate? Well, we're in equilibrium with what? 1, 2, 3, 4, right? We're in equilibrium with the carbamate, right? Now, in that video, I mentioned that we do it with lysine, right? And what if we were reversing it? What would happen? Right? The electrons would kick back in, right? What would happen? This comes out and abstracts a proton, right? So I mentioned that we could do this with lysine, but one of the things you have to understand is you can actually do it with other residues as well. And there's two other points of nucleophilic attack that we can have. Okay, the first one is one that probably when you look at it would make sense, right? What's another residue that has a nitrogen on it? Well, if you said arginine, you're right. So we can actually use arginine, and specifically it's not the nitrogen that has a plus charge, but it's this one. Right? So what happens? If I have carbon dioxide and I load it up in the blood, what happens? Nucleophilic attack. And the mechanism is identical. So you can also have the exact same thing with arginine. So what do you get? Well, you get something that's very similar, right? Very similar indeed, right? Except now the carbamate's attached to what? It's attached to the guanidinium group, right? Remember, arginine's functional group is guanidinium, right? And so now you have a carbamate attached to that, okay? So now there's one other residue we can do this with, and that residue is actually not an actual amino acid residue, but it's if we have the N-terminus. So if I have, and we're assuming it's deprotonated, right? So if I have this set up, right? So here's my protein chain. Of course, here is, this is some R group, right? Some amino acid, right? Here's my N-terminus, right? What happens? Well, if I put carbon dioxide in the vicinity of that, what happens? Well, it's nucleophilic attack, right? On the carbon, pi bond kicks out, what do we get? Well, you get the protein chain, right? So here's my R group, and then this carbamate becomes attached at the N-terminus. And in my biochemistry class, we mentioned that it was at the N-terminus, but realized that anytime you have an amine, this is possible, okay? And generally, the residues that we're talking about where this occurs are lysines and arginines, okay? But as I just mentioned, it can also occur at the N-terminus, okay? So again, remember that by Le Chatelier's principle, if I'm loading up with CO2, there's two places the CO2 can go. Either carbonic anhydrase can consume it to make bicarbonate, or we can attach it to hemoglobin, okay, on these residues right here. And either way, what we're doing is we're solubilizing CO2 and preparing it for transport in the blood. So I hope this video cleared up anything on CO2 transport. See you in the next video.